Okay, so we continue from the previous lessons. When we look at disaccharides, we have done for the formations of the maltose. Then today we're going to look at, in these lessons, we're going to look at this formation of sucrose and then formation of the lactose. And then we're going to discuss, okay, why maltose and also lactose, they are reducing sugar, but sucrose is not reducing sugar. But how we test for this non-reducing sugar, okay? And then we have to refer to the hydrolysis, and we're going to use a maltose as our model to study of the hydrolysis. Okay, so we're going to start first with the formation of the sucrose. So before we have to continue to the formation of the sucrose, okay, we have to note that in order to form a sucrose, we do need alpha glucose, right now, together with the beta fructose. Okay, generally, but it can be alpha glucose with alpha fructose. It doesn't matter, but generally, okay, it will be beta fructose. And we have to look at how the beta fructose look like, and then we will be able to join both of them together. Okay, so let's look at this beta fructose now. Okay, so wait. Okay, now if let us look at this fructose molecule. Okay, look at this fructose molecule. So I'm going to draw the linear form of the fructose molecule. So 6 carbon again. Okay, so for fructose, so 6 carbon. So carbon 1, 2, 3, 4, 5, 6 carbon. And the carbonyl group actually the one that gives the reducing property at carbon number 2. Carbon number 1 is CH2 OH. Okay, and carbon number 3 OH here, and then H. Carbon number 4 OH. Carbon number 5 OH. Carbon number 6 OH. So H, 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 H. Okay, so in exam, you do not need to know the linear structures of the fructose and also the ring structure of the fructose, no need. Okay, so but now what we're going to look at this here is, let me label the carbon. So this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. So the carbonyl group actually at carbon number 2. So therefore, we are going to join carbon 2 and also carbon 5 together. Okay, carbon 2 and carbon 5. So the oxygen atom of carbon 2, oxygen atoms of carbon number 5 will join to the carbon number 2. Carbon number 6 again is not in the ring, okay, again outside of the ring, and also carbon number 1 will be outside of the ring as well. Are you clear? Okay, so now let us look at how we can form this molecule. So first they need to band, okay, they need to band. So this is carbon number 2, carbon number 3, carbon number 4. Okay, this is carbon number 5. Okay, so carbon number 5 connects to carbon number 6. Okay, so look at this. Okay, so where's the carbon number 1? Okay, so carbon number 1 will be, okay, we want to have a beta fructose, so carbon number 1 will be here. Okay, so carbon number 1, look at this. So this group is CH2 OH. So this is carbon number 1. Look at it. This is carbon number 1. This is carbon number 2, carbon number 3, carbon number 4, carbon number 5, and carbon number 6. So carbon number 6, you can see that is again CH2 OH, carbon number 6. So carbon number 5 will join to who? Carbon number 5 will join to the OH and 1H. Carbon number 5. Okay, do you see it? Okay, so what will happen here? Carbon number 4, can you see the carbon number 4? So carbon number 4, we have the OH and H. Carbon number 3, OH, okay, above the ring and H. Carbon number 2, we have the C double bond O. Carbon number 1, you have the CH2OH. So again, I remove the H here. I put a green color. So this H actually can come here. Are you clear? Are you clear? Can I see that? The H, I can move it and then put it here. Okay, so again, let me remove the H and then put it here. So H. So then we're going to form a bond between the O of carbon number 5 and the carbon number 2. And then carbon cannot have 5 bonds, so we remove one of the bonds. Can you see that now the OH group of carbon number 2 above the ring? So this one is the beta fructose. Are you clear? Can you see that? The OH group above the ring. If I put the OH group below the ring of the carbon number 2, then it becomes alpha glucose. Are you clear? Now, why carbon number 2, not carbon number 1? Because the carbonyl groups actually appear in carbon number 2. Can you see that, guys? Are you clear now? Can you see that? It's carbon number 2. So that's why we refer to carbon number 2. But how about glucose? Why alpha glucose, beta glucose, we refer to carbon number 1? Because the carbonyl group in carbon number 1. So this one, carbonyl group at carbon number 2 and OH group above the ring, right? OH group or the carbon number 2 above the ring. So we call it as beta fructose. So simplified form, how we draw it. For simplified form, how we draw this, okay? So very simple. Fructose actually is in the pentagonal shape, not the, okay? It's in pentagonal shape. Are you clear? Not the hexagonal, okay? So how we draw it, so pentagonal shape, we draw pentagon with O here. Can I see that? So carbon number 5, you have the carbon number 6 here, CH2OH. Okay, CH2OH. Can I see that? Okay, with the H. And then carbon number 4, you have the OH group and H. And then carbon number 3, you have the H and OH. And carbon number 2, you have the OH group above the ring and CH2OH below the ring. Can you see that? So this is a beta fructose. So how the alpha fructose look like? So similar alpha fructose. Pentagonal shaped, but CH2OH will be above the ring and OH group below the ring. So that's why this one is alpha fructose. The remaining structure will be the same. Okay, this is a H, OH, H, OH, H, CH2OH. Okay, so can you differentiate between alpha and beta? So I label. So this carbon 1, carbon 2, 3, 4, 5, and 6. Okay, so this carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. So OH group of carbon number 2, if below the ring, is alpha fructose, and OH group of carbon number 2 above the ring will be called as beta fructose. So in order to form the bonds, okay, the glycosidic bonds, then we're going to, react the beta fructose together with, beta fructose together with alpha glucose. Okay, so before we proceed, so in order to form sucrose molecule, to form the glycosidic bonds, we have to react the alpha glucose with beta fructose. Okay, so I'm going to draw the alpha glucose at the top. Okay, so this alpha glucose, the skeletal structure. Now, you should know how to draw for glucose, the alpha glucose, you need to know how to draw. Fructose, no need, okay? So draw the pillar as usual. So this is alpha glucose, so OH group below the ring. Okay, later I will do amendments here. So just for you to see first, H, OH, OH, H, okay, H, OH, H, CH2, OH. Okay, so now in order to form bonds, I put these ones longer. Okay, OH. Okay, I put it longer, easier for me to draw later. Okay, so now I draw fructose. Now, beta fructose. O, pentagonal shape. Okay, so this is a beta. So beta means the OH above the ring with CH2 OH. Okay, then you have the H, OH, H, OH. Okay, so sorry, this is H only, my mistake. And CH2 OH. So I label them, guys. I label them. So this is carbon number 1, carbon number 2, carbon number 3, 4, 5, and 6. For fructose, we have 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. So the reaction will take place between carbon 1 and carbon 2 now. Okay, between carbon 1 and carbon 2. So what actually happened here is one group donates OH, another group donate H atom. Okay, one donate OH, another one donate H. It doesn't matter who donate OH, who donate H in this case. Are you clear? So this one, we write it down, remove as H2O. Are you clear? Carbon number 1 donates OH in this case, and carbon number 2 from fructose donates the hydrogen atom. So you can write down at the side here, this is alpha glucose. This is a beta fructose. And because of this, it's condensation, okay, because remove the water molecule, so it's a condensation process. Then we're going to get a molecules of sucrose, okay, so here we have H, HOH, okay, CH2OH, okay, then draw fructose. So CH2OH, HOH. And the remaining, can you see that the remaining O here, right? So we put it here. So you can see that here we have carbon, oxygen, carbon atom, COC. So this is term F, glycosidic bond. And the reactions are together with H2O release. Okay, so I label this is carbon 1, carbon 2, 3, 4, 5, and carbon 6. So this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. Okay, so I scroll it down. Sorry, I zoom it down so that you can see the overall picture, how the glycosidic bonds can be formed. So in these segments, we are going to talk about how maltose retain its reducing property, but sucrose loses its reducing property. So let me draw out the basic structure of a maltose first. Yeah, so this is a maltose. So maltose, we have two ring structure. So O, two alpha glucose. Okay. Then we have the glycosidic bond formed here. So if you look at this, I labeled this is alpha glucose, alpha glucose. So alpha glucose generally means that you can see that this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6 is outside of the ring. Correct or not? Carbon 6 is outside of the ring. Okay, so again, this carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. Okay, always remember, in order to have the reducing property, depend on two factors here. The first, it must not in ring form. Okay, it must not in ring form. Second, they must have this, what we call the carbonyl group, so that they can have a reducing property. Are you clear? Okay, now we look at the maltose. When the formation of the maltose, the reaction takes place where? Reaction takes place between the OH group of carbon number 1 of 1 alpha glucose and OH group of carbon number 4 for another alpha glucose. Clear or not? Okay, carbon number 1 and carbon number 4. So means that for the first alpha glucose now cannot turn back to the linear form already. Why? Because carbon number 1 is used to form the glycosidic bond. So means that it will remain as a ring structure in aqueous environment. But you look at the second alpha glucose. This part, no bond is formed. Cannot. Glycosidic bond is not formed here, right? So means that the second alpha glucose, that is possible for them to turn into a linear form with the carbonyl group exposed. So means that this part will give us the reducing property later. So means that we can actually make them into a non-ring structure and expose this C double bond O carbonyl group. Are you clear? So what will happen in aqueous medium? You will see something like this. This is the first alpha glucose. Join with the second alpha glucose. But second alpha glucose, you can see that what will happen here is okay, so you can see that this carbon number 1, this is carbon number 1, right? This is carbon number 2, carbon number 3, carbon number 4, carbon number 5, and carbon number 6 is outside of the ring. Clear or not? So this part, the OH, the O just now like this, right? And this one you have the COH, clear or not? So this H remove, come back to this part. By breaking this bond, H will remain going back to here and carbonyl group appear. So this carbonyl group give the reducing property. Are you clear? In Benedict reagent. You get the idea? So it means that we can actually change them back to a non-ring structure. Can you see that? Non-ring, right? This is non-ring structure already. And expose the carbonyl group. So therefore, maltose will still have the reducing property. So that's why if you react the maltose with the Benedict reagent, then what we're going to have? We're going to see the positive color change. It depends on the concentration of the maltose. Then you will be able to see the change from the blue to green to yellow to orange. If concentration is high, you can get the red or even the brick red. Okay, precipitate. Are you clear? So this is about the maltose. Okay. So now look at the sucrose. So we do know that sucrose will react alpha glucose with beta fructose. It doesn't matter beta or alpha fructose, the outcome is still the same. Okay, so here we have the formations of the glycosidic bond. Okay, so if I label here, this is carbon number 1 of the alpha glucose, carbon number 2, carbon number 3, carbon number 4, carbon number 5, where's carbon number 6? outside of the ring, carbon number 6. Clear now? Okay, for fructose, carbon number 1 also outside of the ring. This is carbon number 2, number 3, number 4, number 5. Where's carbon number 6? Again, outside of the ring. This is carbon number 6. So formations of the bond actually take place at carbon number 1 of the alpha glucose, and also carbon number 2 of beta fructose. Then you can see that what actually happened now. Carbon number 1 is the one that have the, in alpha glucose, okay, in the non-ring structure. So carbonyl group actually in carbon 1, the reducing property. For beta fructose, in non-ring structure, carbonyl group actually in carbon 2. So what will happen? Because of the glycosidic bond formed between, okay, can you see that the the glycosidic bond formed between these? Okay, let me show you. Okay, can you see my pointer here? Okay, now you can see that now carbon number 1 is the one that gives us the reducing property, right? Because the carbonyl group in carbon number 1. And carbon number 2 for the beta fructose carbonyl group at carbon 2. So what will happen now? Because they form the bond like this, can I make them into the non-ring structure? The answer is no. In aqueous now, you can see that because the formation of the bond involves the carbon number 1 of the alpha glucose and carbon number 2 of the beta fructose, therefore, sucrose now cannot turn into the non-ring structure. Therefore, they lose the reducing property now. Can you see that? Why? Because the formations of the bond, if the question asks why reducing sugar, the sucrose is a non-reducing sugar, because formations of this glycosidic bond. Okay, the answer involve. Okay, carbon number 1. Okay, the OH group, sorry, more in detail. Okay, OH group of carbon number 1 in alpha glucose and OH group of carbon number 2 in beta fructose. So therefore, now they lose the reducing property. Because they cannot turn into the linear structure anymore. Can you see that? How we turn? Can you see that? Okay. Are you clear? So look at the structure now. And compared to the maltose, maltose at least we have one side that still can have the reducing properties. Okay. So in Benedict, okay, reagent, when you do this, the non-reducing sugar test. In non-reducing sugar test, what we do, okay, still remember, our process to in order to test the non-reducing sugar is not a direct. We don't have a specific test to test the non-reducing sugar. So what we do? So first step, we always carry out the reducing sugar test first. If we use the reducing sugar test, what is the outcome? Because if the sample only contains sucrose, and sucrose is a non-reducing sugar, you can see that there is no change in the color. So it means that remain blue. Correct or not? Because the sucrose, what is the conclusion? Because the sucrose, okay, is a non-reducing sugar. The explanation. Okay, because, okay, sucrose is a non-reducing sugar. So therefore, okay, what is our conclusion? Because it remain blue, right? Okay, so remain blue, then therefore we claim that reducing sugar absent. Correct or not? So now we say first, we say that, okay, confirm, reducing sugar absent already. Okay, so now second step, what we do? We take a specimen and we do the hydrolysis. So how we carry out hydrolysis? So we add dilute HCl. Correct or not? And then we heat it in the boiling water bath for 3 minutes. So in this process, what actually happened? In this process, we can see that now sucrose is hydrolyzed into a glucose plus fructose. So means that if you look at the ring structure of glucose now can turn into a non-ring structure. So with the carbon here, and because it's the alpha glucose, so below the ring, C double bond O still here. Can you see that non-ring structure? Fructose also. So carbon number 2, okay, this is carbon number 2, this is carbon number 1. So carbon number 2 of the fructose now again can actually have the carbonyl group. So for the glucose at the carbon number 1, this carbonyl group appear. So both of them, they are reducing sugar. Correct or not? Okay, can you see that? Non-ring structures, right? The glucose can turn into non-ring structure, fructose can turn into the non-ring structure. But sucrose, too bad, cannot. Oops, sorry. You see that we join this way, right? Okay, the oxygen here. So therefore, you can see that this part carbon number 1, this is carbon number 2. So no way for them to detach. Can you see that? Are you clear? Unless we carry out the hydrolysis. And then after that, we add the Benedict and the solutions. Again, we heat at boiling water bath for 3 minutes. Are you clear? Oh yeah, sorry. More precise, then we have to add one more step here. Sorry, guys. What is the step? Okay, so before that, we need to have a neutralizations, okay, with sodium bicarbonate. So what is the functions of this? Because we want to remove, okay, HCl or neutralize the HCl, because the Benedict reagent work the best, okay, at alkaline conditions. Okay, so we add bit by bit until no bubbling process take place. Then we add the Benedict solutions, we heat it, okay, at the boiling water bath for 3 minutes. So if the solutions remain blue color, so then we can conclude that non-reducing sugar absent. Correct or not? Okay, based on the first one, blue, second one also blue, then we can conclude that non-reducing sugar absent, or we say sucrose absent. But if you have the series of color change now from green, okay, to yellow, okay, from yellow, let's say change to orange, orange change to red or bright red, then what is the conclusion? We can conclude that non-reducing sugar present. Okay, but before we make this judgment or conclusion, you need to check back the first results here. Are you clear? If they still remain blue, then therefore means that at first there is no reducing sugar, but after that reducing sugar presence, right? So make sure that now we can conclude non-reducing sugar or sucrose present. Okay. As we know that in our syllabus, you are not required to know how the formations of the lactose take place. But I will still show you guys how the lactose take place, because question can give you, okay, question can give you, I mean, the structure of the galactose and ask you to actually form the bond. Okay, so in order to form the lactose molecule, okay, so how we use, again, we need alpha glucose and beta galactose. Okay, alpha glucose and beta galactose. So I'm going to draw out the structure. So later you can actually copy it down inside this box. Okay, let us compare the structure first between the glucose and also the galactose. Okay, glucose and galactose. So first, let us look at the alpha glucose. Okay, a simplified structure of alpha glucose. So O, the alpha glucose is the one that we need to know how to draw. So that's why I keep on drawing the alpha glucose for you guys so that you guys can remember. Okay, so CH2OH, H, OH, 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 because alpha glucose, so OH group below the ring. So this is alpha glucose. Okay, so now let us look at what is the main difference between the alpha glucose and also the alpha galactose first. Okay, this alpha galactose. The main difference of alpha galactose basically at carbon number 4. So this is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and carbon number 6. So alpha galactose actually have a similar shape as the alpha glucose. When you draw it, so CH2OH, H, so OH, OH, so HOH. So the one that make them different actually is at carbon number 4 where there is opposites. Okay, the rotations here, can you see that at carbon number 4? If you compare alpha galactose and alpha glucose. Do you get it? Can you see that? Okay, so what is the beta galactose? You look at the beta galactose. Yeah. Can get it now. So you compare alpha galactose and beta galactose now, easier for you to compare also at carbon number 1, but OH group now above the ring for beta galactose. So OH group now above the ring. Okay, so H, OH, so OH, H. So carbon number 4, always remember, is OH, and carbon number 5 and carbon number 6, okay, as usual. So this is beta galactose, okay? So carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. So why? Why alpha? Because OH group below the ring, okay? In this case, why beta? Because OH group above the ring. I didn't record, so now I record again, sorry. So I repeat, guys, so this is beta galactose where the OH group above the ring at the carbon number 1, okay, and alpha glucose OH group below the ring. So if you put them side by side, can you see that one OH group above the ring and this OH group below the ring? So we cannot form the bond. Can you see that we cannot form the bonds? So what we need to do, we need to rotate the structure 180 degree. Okay, can you see that? I rotate the structure 180 degree upside down. So it means that OH group initially above the ring. Now can you see that below the ring? Can you see that? So O at the back, then we flip it, it become at the front. So CH2OH below the ring now, OH group above the ring, OH group below the ring. So everything upside down. So that now OH group and OH group, can you see that? So this is a beta galactose OH group. Okay, with the alpha glucose OH group, now they are side by side. We can form the glycosidic bonds by removing the water molecules. Can you see that? When you remove the water molecule, we can form the glycosidic bonds. So can you see that? So after the condensation process, we will form the glycosidic bonds. The bond will join carbon number 1 of the beta galactose and carbon number 4 or alpha glucose. Can you see that? Are you clear? So why lactose remains the reducing property or retain the reducing property? Why? Because this part, no choice, because it's formed the glycosidic bonds. But this one, yes, they can actually go back to the non-ring structure. Okay, so how it look like? So it would be something like this. So carbon number 1. So carbon number 1, the OH group, right? So now H put back here. Can I see that? Carbon number 5. Can I see that? Okay, so this is glycosidic bond. So you can see that it's carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So carbon 6 is actually outside the ring. So this part loose. Can I see that? This one no longer have reducing property already. But if you look at this, this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. And now you have carbon 6 here. So this part actually, can you see that? Carbonyl group is still here. So this is a non-ring structure in the aqueous. And you can see that the presence of the carbonyl group gives us the reducing property. Okay, so you need to copy, take a photo, you can. So first part, you can compare between the beta glucose and alpha glucose first. Okay, you can take a photo. So how we rotate it 180 degree upside down. And then how the formations of the bond take place. Take the photo if you need. And last, how comes the lactose still can retain the reducing property. Okay. So whatever we have discussed just now, okay, when we join two monosaccharide together, they're going to form the glycosidic bonds, okay? The glycosidic bond, we're going to join them together to form the disaccharides. So the reverse process is termed as hydrolysis, okay? The reverse process. So we're going to use a maltose as one of the model to study how the hydrolysis actually take place and how we show in our drawing the hydrolysis process, okay? So let's look at the hydrolysis process. When we have one molecule of maltose. Okay, let me complete this one first. So maltose we know is alpha and alpha. So we complete diagram CH2OH, H, HOH, OH, H. So alpha, right? So OH below the rings. Okay, so glycosidic bond here. Okay, so this is a molecule of maltose. Okay, this is a molecule of maltose. Okay, so what we're going to do here is we're going to carry out the hydrolysis. Okay, so how to carry out hydrolysis, how to show it. So plus water. So how we plus the water? So I would normally do this. I will write here, okay, to bracket out COC, I will say that glycosidic bond is broken by H2O or water molecule. Are you clear? Okay, so it plus H2O. So it indicates that where is H2O? H2O actually will break this glycosidic bond, or more precisely, alpha 1,4 glycosidic bonds. Okay, so what is the outcome? We're going to form 2 glucose, but you have to separate 2 glucose, yeah? You have to separate 2 glucose. So O. So CH2OH, H, HOH, HOH, HOH. Okay, in the show. And another molecule. I will tell you guys why we need to draw 2 times. In fact, they are the same thing. Then I will do this. From H2O. Okay, so this is how you show the hydrolysis of a maltose. Okay, so this is a maltose. Hydrolysis of maltose will produce 2 molecules of glucose, or more precisely 2 molecules of alpha glucose. Okay, so you can take a photo for this, and we have done for disaccharides.